a slide that is very close to my heart. It's actually a quote from Charles Darwin's book, which you may have heard of, The Origin of Species, 1859. It says, "Endless forms, most beautiful and most wonderful, have been and are being evolved." And I think Darwin tells us about the mechanisms of how we get species, and his book is great. It's a fun read, but what it doesn't tell us is what those differences actually are. How do we get the differences between, for example, these beetles here? They're all different colors, they're all different shapes and sizes. We know these are closely related beetles. They all come from a common ancestor, but somehow they've morphed into these different forms. And Charles Darwin tells us how that happens in terms of what the mechanism of speciation is, but he doesn't tell us what makes the difference. What is it that makes that beetle green or this beetle rainbow colored? And I think that's a really important question, and one that I've been trying to answer all my career. So I'm going to start by talking about that, and then we might go into some weird places, though the fact it's messed up my text already suggests that this is going to be a fun talk. The mechanisms, we assume that the mechanisms that make the difference between these species are the same mechanisms that make the difference between these guys. These are all animal phyla, right? So these are all the different sort of major groups of animals on Earth. I know a lot of them look like worms, but some of them are familiar. I've put a little mouse there, so there are vertebrates, there are insects, there are various other things. And one of the cool things about the variation in animals is it's non-continuous. So if we believe that there's a sort of these mechanisms which can make any form we like, when we look at this higher level organization, that's not true. So for example, all insects have 6 legs. There are no insects with 8 legs, but there are spiders. They've got 8 legs, but none of them have got 6 legs. All vertebrates have got 4 legs, which is why flying horses are such a problem, right? So these kind of constraints are interesting, and it suggests to me that the way we make an animal somehow constrains what that animal could look like, so what the potential of an animal form might be. But this wasn't always the case. If we go back, as I'm sure you will, 550 million years, there are fossils of things that are very like things that are alive today. So this thing down here, that's quite like Neil Gemmell. That's a chordate, right? That is your ancestor in the fossil record 550 million years ago. This thing up here, that's a peripatus, right? It's living on my thing, and it's living on the hills around Wellington. Looks exactly like that, except that one is marine. It's weird. But this thing here, it's a sort of shrimpy thing, except not very shrimpy thing. It's got 5 eyes and a trunk. There's nothing like that on Earth today. So somewhere in the history of life, our ability to produce all of these different forms has been constrained. We no longer can make things with 5 eyes in the trunk. We can just make more insects, more beetles, right? So if we want to understand how diversity is constrained, I think we have to start by asking the question, how is diversity built? And so I am always very interested in embryos. So this is a movie of a single Drosophila embryo. There's just a bunch of them. So Drosophila is a fruit fly, and this embryo is undergoing a process called gastrulation. You can see the tissues moving. You can see this coordinated movement of stuff that's going into the embryo that's going to form the gut. That happens in all animals. Gastrulation, that is in fact the most dangerous point in any human's life, right? A third of embryos, human embryos, don't make it through gastrulation. So these sorts of movements of cells which take a single fertilized egg and produce an adult organism are controlled by something. And the something they're controlled by are genes. Genes sitting in the genome of the species. Right? So, I pressed the button. So if we want to understand how you make an animal from a single fertilized egg, and then how that changes to produce different forms of animals, we have to look at the genes. We have to look at their genetics. So if we sort of been doing this for a long time, let's sort of look at 2 examples here. At the top here we have a fruit fly embryo. At the bottom we have a bee embryo. These embryos have been stained with stuff and beaten about, but this green staining here is a gene called even skipped. It's present in both the Drosophila and in the honeybee. These 2 embryos are 300 million years derived. It's another 500 million years to a human being, which also has an even skipped gene. Right? This gene in Drosophila marks out the developing segments. It does the same thing in honeybees. It doesn't do that in humans, so we'll just ignore that. But what that means is that this change in form of these embryos to produce different forms of life is not new genes evolving or new processes evolving. It's taking genes that are already sitting in the genome and changing how they're used. It's not what genes you have, it's what you do with them. So my entire career has been trying to work out how the way we use genes changes, okay, over time. And the first thing that really became obvious to me is that if we want to understand how genes change over time, we need to understand what genes there are in an organism and how they are regulated. And to do that, the first thing you have to do is sequence a genome. And so I've been incredibly privileged to live at a time where my science went from cloning a few genes, and I can see Tony there holding his head going, yeah, he knows this thing. There was this moment in biology where we went from, oh yeah, I can look at a gene, that's great, to, oh my God, what is a genome? What's in it? I've got to worry about 20,000 genes, not one. It's been great to live at that time, and it meant that I got involved in things like sequencing the honeybee genome, which was the third insect genome to be sequenced, the 8th animal. It's quite a remarkable genome. It's fun, but that genome contains all the information, or at least we think it contains all the information, to make this. This is a remarkable organism, right? It's got arms and legs, it's got eyes, it's got antenna, it's got behaviors. All of that somehow is encoded in its genome. And I've only got 5 minutes to go, and we've got about 20 slides. I'm just going to have to go faster, if that's possible. All right, but the cool thing is, so I spent a lot of time trying to look at honeybees and compare it with fruit flies, which are the geneticist model, which was the first insect sequence. We could look at how things have changed over time, but actually bees are even more interesting than that, because bees do this remarkable thing in that they produce 2 different animals from the same genome. Those 2 different animals are on the slide here. So this one here is a queen bee, and these guys are workers. And you can see the queen bee is larger, but it also has a different brain. Its brain is wired differently. It produces different pheromones. It does all kinds of remarkable things. In fact, on that slide, you can see this happening because all of these bees, the worker bees, are orienting to the queen. They're all pointing at her, apart from the dumb one up here. All right? All of these guys are pointing at the queen because she's expressing pheromones which control and produce the activity of the hive. So we spend a lot of time trying to understand how genes are regulated differently when you're developing a queen bee versus a worker bee. So that's all great, and the government would say to you at this point, fundamental research, Peter, it's not terribly useful. Great, how does this help us? The answer is, this helps us enormously. So we took this kind of knowledge and the techniques and the ideas we developed in this thing, turned it into a 5-year program trying to understand and improve bee breeding in New Zealand. New Zealand's beekeeping industry is a $100 million industry a year. It is on its last legs. It's really suffering, and one of the problems is that bee breeding is really, really hard. So we developed the statistical techniques, the assessment techniques, and the mathematics and biology required to improve bees in this country in the hope that that bee industry would take it up. But working in insects, you sort of realize that after a while, people have 2 views of insects. They like fuzzy ones, and they hate stingy ones, right? And New Zealand has a problem, right? So we have a beekeeping industry we want to look after. We also have a load of invasive wasps. These wasps come from Europe. They have the highest numbers in the world in New Zealand. They cause $1 million of traffic accidents a year. They damage our economy. They damage our primary production, and they steal kids' jam sandwiches at school. So people want us to kill wasps. How do we do that? We take exactly the same approach we have. If we can understand how genes control what a bee looks like, how it behaves, how it interacts with its brothers and sisters, mainly sisters, we can also look at the same thing in insects like wasps, invasive insects, and find the chinks in their armor that we can use to control them. And so we're now developing genetic technologies from the same sort of systems to control wasps in this country, in the hope that we can save this country what is $220 million worth of wasp control a year. Okay, so you're all sitting here going, oh, he's really keen on insects, isn't he? But we use the same technologies, same ideas to look after these fellas. So that is a kakapo. That's my favorite kakapo, Zephyr. She bit me. We sequenced the genomes of every single living kakapo and are just sequencing the 104 chicks that have just been born, which is fantastic, and use that info. Oh, I'm going to show the video again, are we? No, we're not. We're going to show penguins. Oh, it's not working. Go. Oh well, you get the penguins. Using the same kind of approach as linking the genetics, the gene regulatory networks in these species to understand how they respond to the environment, and to take that information to develop tools that the Department of Conservation can use to better look after the species and to improve breeding and to improve our kakapo populations. And I can't claim credit for it, but 104 chicks this year is pretty damn impressive, right? That's almost a third more of the population. We're doing similar things with hoiho, the yellow-eyed penguin. This is affected by a fairly horrific virus, but we know there are resistant members of the species, and so we're trying to find why they're resistant and see whether we can use that to help breed more resistant penguins, or whether to think about how that might be interacting with the virus. We also, and just sent a report to MPI this morning, have been working on hornets. Those of you who live in Auckland will know you've been attacked by giant invasive hornets. You'll be glad to know we've sequenced every single hornet found in this country, and we can tell you stuff about them. Unfortunately, I have an NDA with MPI, so I can't tell you anything about them except they're really nice. 1 minute, man. But at least I get to show you this, which is the fact that even though we're doing these applied stuff, we have to go back to that well of fundamental research. This is the embryo of a peripatus, that thing that I showed you in the fossil record 550 million years ago. This one is stained for a couple of genes which control leg development, because we're trying to understand how their legs evolved and developed from their ancestors. So they're blue staining. This image came from one of my students 2 days ago, and I couldn't be more proud to be working on a species like that. I'm going to very briefly say we also do stuff for kids, right? We have a thing called Lab in a Box to go to rural communities and go and teach them how to put on bee suits and how to get stung by bees and why bees are important. Finally, I'd just like to say thank you. At this point, most scientists come up with the acknowledgement slide with 1 million people on it. I couldn't fit the 1 million people on, so I'd just like to apologise to all the people that I've worked with and hope that you're all right now. And I'm funded by Genomics Aotearoa, the Ministry of Business, Innovation and Employment, Bioprotection Aotearoa, and brought to you by University of Otago, Otago Waka Ihu Waka, and the number 7. Thank you very much.